Good afternoon, everybody. So first, thank the organizer and the Singer Institute for providing this opportunity for me and my work and dancing here today. And my topic is alternative evolutionary endpoints for hypoxic regulation of Candida glabrata and the related species. So by looking at the phylogenetics tree for Saccharomyces, we can see a lot of that indicates pathogenic and that indicates non-pathogenic. So in Candida clade, we can see a lot of pathogens, so like the superstar and the albicans. And my project is looking at the Saccharomyces clade, so Candida glabrata, which is the second or third cause of Candida infections. So it can grow the pseudohybrid to form the biofilm infection. And another two related species, Saccharomyces cerevisiae and Glabrata. So by using this comparative analysis of different fungal genome to exploring the evolution of transcriptional regulation, particularly to the hypoxic response and drug resistance. So previous work shows us have the Saccharomyces cerevisiae. There are two pathways here. The first one is heme pathway. So HEP1 is a major regulator to regulate the ROX1, which is the repressor of the hypoxic genes. And the second pathway is the sterol pathway. So UPC2 is regulated to regulate the Gostra family genes in hypoxia, and the ECM tended to take over the function in normalcia. So first, let's look at the ROX family genes. So there are three major groups of ROX family genes. So ROX1, ROX2, and the IFG1. In Candida clade, so they lost the ROX1 and they have the IFG1, which is because and the previous work showed so it loses the hypoxic regulation, it regulates the filamentous growth. And in Saccharomyces clade, so there are three species. So some species lost ROX2, so like Saccharomyces cerevisiae, and some species lost ROX1, so it contains ROX2, and like Candida glabrata and the Pneumocystis castalia contains both of these two genes. So we carried out the microarray and showed and we investigated the hypoxic response for Candida glabrata, which is very similar to most of other fungi. And we compared the wild type hypoxia profile and the ROX2 pneumonia profile, and we found 89 overlap differential expressed genes is in upregulation of sterol mechanism, sterol genes and heme13. So which suggests the ROX2 in Candida glabrata is the repressor of the hypoxic genes. And then we use the same assay to identify the hypoxic response for Castalia, so which is very unusual. So there is no increased genes and 91 decreased genes. And also then we use the copochloride to mimic the hypoxia, and which we found the copochloride can induce the increased expression of sterol biosynthesis and the heme biosynthesis, and decrease the ribosome biosynthesis. And some are different, so it's the copochloride can induce the increased expression in respiration. And from the so for the phenotype screening, and we found the Saccharomyces cerevisiae ROX1 deletion and the Candida glabrata ROX2 deletion, and the Pneumocystis castalia ROX1 deletion phenotype in the presence of copochloride. And also we transformed the Castalia ROX1 and the ROX2 gene to Saccharomyces cerevisiae and the glabrata deletion. What we found is only Castalia ROX1 can complement the phenotype in cerevisiae, and both of these Castalia 2 genes can complement the phenotype in Candida glabrata. And the second project is looking at the UPC2 paralogs. So in Candida clade, so it only has a UPC2, but in Saccharomyces clade, like these three species, so it has both UPC2 and the ECM22 paralogs. So in Saccharomyces cerevisiae, we know UPC2 regulates the hypoxic genes. And what we found is in Candida clade, so UPC2 is the major hypoxic regulator, but in Saccharomyces cerevisiae, so UPC2 regulates hypoxic response as well. But in Candida glabrata and Castalia, so the UPC2 and the ECM22 swap the functions, and the ECM22 is the major regulator in hypoxia. And also we use the copochloride to mimic hypoxia. What we found is UPC2 and the ECM22 are both the regulate the mimicking, have the co-coagulated response. So that brings to my summary. So the hypoxic response to Candida glabrata is shared with other fungi, and in Saccharomyces clade, the ROX1 is a major regulator, and the ROX2 take over the functions in Candida glabrata. So Castalia also has a very unusual hypoxic response, but it shares the increased expression of other genes, heme genes, with Candida glabrata in copper chloride. And the role of ROX2 in Castalia is not very clear, but the transcription profile suggests that ROX2 regulates the growth. And also, we always express ROX2 in Castalia, so it has a phenotype in Congo red, so which suggests the ROX2 in Castalia, like the gongo in the church, protect the cells. And the UPC2 is a major regulator of the steroid levels in the CTG-related Candida species. And the Saccharomyces ricei and the ECM22 perform the role in Candida glabrata and Castalia. So I would like to thank you, my supervisor Jerry Butler, and the rest of members in our lab and our collaborators from Conway group and Ted White group.
Textbook timekeeping there.